The overall goal of this procedure is to monitor prion-like spreading of protein aggregates in vivo, using transgenic flies that express an aggregation-prone protein and a soluble version of the same protein in isolated cell populations.
This method can help answer key questions in the cell biology and neurodegeneration fields, such as how protein aggregate pathology or other material transfers from cell to cell in the brain. The main advantage of this technique is that it allows for imaging of protein aggregate spreading from one cell cytoplasm to another in an intact central nervous system.
The implications of this technique extend toward therapy of Huntington's disease and other prion-like neurodegenerative diseases, because aggregate spreading appears to be a common mechanism underlying the progression of these fatal disorders.
Using standard culturing conditions, generate transgenic Drosophila melanogaster lines containing tissue-specific Gal4 or QF drivers, as well as lines containing wild-type or mutant Huntington transgenes downstream of the Gal4 UAS or QF QUAS. Collect the progeny of the desired genotype and age them until they are ready to dissect. To prepare for the brain dissection, collect the solutions needed and chill them on ice, along with a glass multi-well dish, pipette tips, and 1 pair of forceps. Next, anesthetize the adult flies using carbon dioxide and transfer them into a well of the glass dish. Then, add about 1/2 milliliter of PBST to the well with the flies and to an adjacent well. Next, under a dissect